Start by saying bad news. The test has been moved back to Friday. Why? Bull crap. Must be bad news.
I want my test.
Hey, wait a minute. Friday? When was it supposed to be scheduled for? Wednesday.
Monday.
Next. You don't have.
No, that's right. It's Friday. All right. Okay. Any questions?
The week after. I think that's
Any questions?
You want to have class Monday?
No, that's the following week. Following week, we're squeezed between. We got one day of class the following week. We don't have Monday. We don't have Wednesday.
So, well, math and biology still
I don't want you waiting till the end.
well, right, but it's just not next week.
No good deed ever goes unpunished. I never thought of that one.
I did.
Well, how about this? I'll send out a message if it changes. If I get it, the question was, is mastering biology closing date going to be pushed back then? Since it was closing the night before, which was Tuesday, could I move it back to Thursday? It's only 2 days difference, and I prefer you did it before that anyway. But, all right, I will strongly consider it. Let's put it that way. I will strongly consider it. And if I do, I will send out an email just saying I have changed the date. And again, let me know if there's a scheduling conflict. Let me know. Yes.
What is the difference between the nucleus and the nucleolus?
Nucleus and nucleolus. All right. Nucleus is that membrane-bound package in the cell that contains the DNA. And it is, remember, it's not just covered by membrane, it's covered by a double membrane nuclear envelope, right? A nucleolus is a mini nucleus inside the nucleus. And what? Correct. It's actually just a darker area where parts of ribosomes are being put together. So if this was the cell, and this is, you wouldn't see the double nucleus. What you'd see is just one thing. But if you look closely, you'd see like a darker area here and a darker area there. There's really no limit to how many, but that's the nucleolus, the site of production of ribosomal parts. Now, once the cell begins to divide, the first thing that happens is the DNA begins to wind itself up. They disappear. So you only see those in chromatin. So they're not a thing. They're more of a region. It's kind of like saying where's it pointing to a shadow. The shadow is only there because something else is going on, right? So in this case, you're seeing ribosomal parts being constructed, but it would only do that when the cell is, when the DNA is all unraveled. So nucleoli are inside of the nucleus. Okay?
This is going
Sure.
back, but what's the difference between like excrete and secrete and the
Purpose. Purpose. Secrete is to be given off for a reason. Excrete is to be given off just to get rid of it.
what's absorbing?
That's, you can do a number of things. It can be pinocytosis. It's endocytosis. Again, we don't ever absorb anything unintentionally. Well, we do, but usually that would be diffusion. So there really isn't a term other than absorbing. Sometimes, we also use the term resorbing when we're moving it from one part of the body to another. So, anyway, good questions. Anybody else? Yes, no? No? Okay. All right. Wrong one. Doing AP2.
Hey, I got a quick question.
Sure.
The chromatin is when it is all
Other way around.
That's when it's straightened out.
Right.
And then it's chromosome when it's all straightened out.
Correct. His question was the difference between when you're describing DNA, when do you use chromatin and when do you use chromosome? Chromosome can be used to explain a single molecule, whatever form it's in, right? So we have 46 chromosomes all the time, other than egg and sperm. However, if you're talking about their condition at any given time, when they are wound up tightly into solid structures during division, we say those are chromosomes. When they are unraveled into a pile, we call that chromatin because we can't visually separate one from the other.
So is that when they start to divide and they pull apart? Is that those are chromatin?
No, it's the other way around. When cells divide, it would be in the form of chromosomes. They're more easily managed. Think of it like the balls of yarn. If we were going to separate, if we had 46 balls of yarn in a pile, and we were going to separate them into 23 and 23, you'd wind them up first, it'd be much easier. Right?
Yep.
Yeah, so that's kind of a way to look at it. All right? So really what happens is, as it starts to wind up, we lose the nucleoli first, because once they start winding it up, that DNA can't be used to make ribosomal parts. All right, so then they start to, they condense down to solid structures, we split them, we make some new membrane, split the cell in half, and we just start over again. The DNA unravels back to chromatin, and then, okay. So, we were talking about, we did finish up the cell, didn't we? The cell parts?
Yep.
All right. I was going to ask one question at the end, and that was, can't think of it right now. So, we'll go back to cell membrane. Now, you are going to get tired of hearing this, right? But membrane is membrane is membrane. It's kind of like saying one type of soup has the same broth as another, has another, has another. They're different soups because they have different things added to it. But the broth of the soup is the same. So, membrane that we start off with, the outer cell membrane is called the plasma membrane. I learned it as the plasmalemma, but we call it the cell membrane or the plasma membrane, and it literally defines the outer covering of the cell, right? I mean, that's the outer edge. It's like the fence on a field. The outer fence defines the field. If you're a farmer, you have fence all the way around your field, keep your cows in, or whatever animals you're raising, okay? Now, the one thing we have to remember about the cell membrane is just like a fence or a gate, it allows certain things in, but keeps other things out, right? Let's say chain link, a very common type of fence, right? Animals that are small enough to crawl through or crawl under the fence can get through, but larger animals that can't get through or over are kept out, correct? So is a fence selectively permeable? Yes. Certain things can get in, certain things cannot. So we use the two terms, selectively permeable or semi-permeable. That means that some things can pass through and others can't. Now, let's quickly go back to the whole why are cells small issue, right? Why are cells small? What's the surface area issue? We want
greatest surface area for the smallest amount.
right, we want the most surface area per volume. So we want a high surface area to volume ratio, and you only get a high ratio when the objects are smaller. All right, and that's because we want membrane to communicate with the outside world, right? But if that's the case, membrane regulates what goes in and out. So isn't membrane kind of running the show? Almost, kind of thing. I mean, the nucleus has all the DNA in it, but really, the nucleus and all those internal parts can only do what they're allowed to do. They can only do how much oxygen they have brought in from the outside, with the amount of fuel they have brought in from the outside, with the amount of waste that's given off to the outside. So you see why I say the membrane kind of runs the operation? So the cell is strongly dependent on the membrane allowing in what they need and giving off what they don't need. Okay. So this is the model that we usually use. This is the one we get in our book. Now, doesn't that look like couldn't that be a decent puzzle? No, seriously, I'm saying that there's parts, right? I mean, you ever look at a cake and try to think what's in a cake? Could you separate the pieces? Can you take the eggs out of a cake? Or the sugar out of a cake? No. But could you take the proteins or the lipids out of this? Yeah. So that's why we call it a fluid mosaic model. Fluid in the sense that these parts are not glued together. They're not covalently bonded like some giant polymer. They're actually stuck together sort of like magnets. You know, you can pull them apart, stick them back, pull them apart, stick them back, right? And in fact, the pieces of membrane can move side to side very rapidly. The numbers are astronomical, how many times these things change per second. So if you think about it, what's happening? They're swirling. They're moving around. And all these components are. Very rarely do they flip sides. But they still do. All right? That's why we call it fluid. And mosaic is the sense that they have many pieces. Has anyone ever seen a true mosaic piece of art? There's a church in a cemetery in Minneapolis. The whole inside is done in little tiles. It's called Byzantine mosaic. It's a very common art form from the Middle East, like a few hundred years ago. Beautiful. But it's all done in little tiles about this big. Same thing with the membrane. If we go back and look at it, think mosaic. Doesn't that sort of look like it? All these tiny pieces, and they're all able to move around each other. So that's the whole idea we need to understand as far as what a membrane, how it functions. Because they're not stuck together, we can separate them. We can also put them back together again. But, is there variation? Yes, there is. And the variation primarily comes from the proteins. Okay? Membrane from one cell is very similar to membrane from another cell. But, because one cell has a different function than another cell, it probably comes from, partially from the proteins in their membranes and the other things that are inside. All right, so just because we say it has protein doesn't mean they're the same proteins. So, first of all, the primary core of the membrane itself, we talked about this when we looked at the macromolecules, right? Phospholipid. It's got two legs that are fatty acids. It's got a glycerol in the middle, a phosphate, and probably something like a choline or an inositol at the top. So the bottom part of the molecule here is going to be hydrophobic. The top part is hydrophilic, meaning the top part likes water, the bottom part doesn't. And by doing so, we now have an amphipathic molecule. And when you put them together, particularly in an aqueous environment, which is the way most living things are, they will form sheets like this, either little spheres called micelles, or these sheets of tissue like this. So now we'll start with that, and then we'll build our membrane from there. So, you notice, because of the molecules being amphipathic, the membranes then take on those qualities. Okay? For instance, one of the emerging qualities here is the fact that the outer layers are hydrophilic and the core is hydrophobic. So imagine this, something as simple as a molecule of water. Everything likes water, right? But that water molecule comes in and it approaches the hydrophilic end and, hey, you liking this? It's attracted to it. All of a sudden, it gets through here and runs into this. This is the hydrophobic core, and it's actually repelled. In fact, any molecule that's polar is going to be repelled by that hydrophobic core. So, you think sugars would pass through that very easily? No. Anything that would dissolve in water very easily would not pass through lipid membranes. And that plays a big role in your health, because any hormones that are protein need another mechanism. Steroids are what type of molecule? Cholesterol? What is cholesterol categorized as? A lipid. Would a lipid pass through that? Yes, and quite easily. So in fact, that's how steroid hormones work. They don't have to knock at the door. They got one of those open door policies. They just shoot right through the membrane because it's all the same. So they allow like molecules to pass through. So that's the first thing. We now know that lipids and nonpolar molecules can pass through very easily. Nonpolar molecules cannot. So in that case, now we got to figure out what can we do then? Because we can't live without nonpolar. We can't live on nonpolar molecules alone. Oh, and by the way, it also explains something. Why chemicals like gasoline are so dangerous. First,
Because they can pass through.
it can pass through, and sometimes even worse than that, it can dissolve your membrane. Yeah, solubilize your membrane, and there you go. So those things, you think about it, a lot of the products in gasoline are quite toxic. So anyway, let's add a little bit of cholesterol now. We talked about that. And because cholesterol is also amphipathic, it's going to line up nice and neat. And you remember, what's the purpose of cholesterol? It's a stabilizer. It's a stabilizer at extreme temps. So, think about it. Lipids, right? When you heat lipids, what tends to happen to them? They liquefy, right? Be it butter, be it shortening, even oils will thin out as you heat them, okay? So, by adding a little bit of cholesterol, it stabilizes the membrane at high temperatures. So basically it's saying it doesn't melt as fast. On the other hand, when they get to a low temperature, they would actually solidify. The fact that cholesterol is in there means they stay fluid to lower temperatures than they normally would. So it's an antifreeze as well. So cholesterol gives some stability at extremes that membrane without cholesterol would not. Okay? So again, it's a moderation thing. Too much and you become too solid, too little and you become not fluid enough. All right?
So would that go to say like lipids from animals that are already saturated, if they had high cholesterol, they would be...
Okay, you're talking about two different issues. You're talking about cholesterol and saturated fats. Both are lipids and both have negative side effects. And it's because animal fats tend to be saturated, which means they tend to have a lower melting temperature. Sorry, no, a lower freezing temperature. They'll solidify faster. So they also tend to fall out of solution faster and they can cause plaques and things like that. Cholesterol is similar. Cholesterol has a lot of negative side effects when we have too much of it.
So the cholesterol is attracted to the middle. How come it's not repelled by the polar heads when it comes to cholesterol?
Because it has its own polar head.
Oh.
It is amphipathic. It has this tiny little tail on it sometimes they do have hydrophobic tails, but the end will also have like a hydroxide on it. So one end will be hydrophobic, the other one will be hydrophilic.
So non-polar molecules can't pass through? You think that you would have
Non-polar molecules are what would pass through. Polar molecules do not.
How does it get past the polar head if it's not polar?
Only the end is. Okay, I know what you're saying.
Yeah.
It's kind of in the formation of these things. They start with just kind of a pool. And now exactly how it's built is a bit more complex than that. But it's kind of like you just sort of, they fit together or they fit as you're building it. You just keep lining them up the right way.
Yeah.
Cholesterol just fits in there with, like the pink one, there's cholesterol.
Okay.
Like that. Because in fact, when they're building these things, they probably don't even recognize the difference because they're both amphipathic. So if you got a bunch of these things together, they just all come together, and since they're fluid, they can move around, so they don't have a particular position to be in.
So the non-polar, non-amphipathic ones, they just kind of sneak in?
There really wouldn't be. Anything that's polar, you're talking about like the steroids, that's a bit more complex, but like I said, these are only held together so tight. It's not a major barrier. And how they actually get through, that's a great question.
Okay.
Yeah, they would have to get through, but remember, they're not glued together, so they're not permanent. So they would actually be attracted to the middle part of the membrane. It's called a hydrophobic interaction, where hydrophobic molecules are actually attracted to each other. So it would actually pull.
That's greater than the repel from the polar.
Yeah. So anyway, but that's a great question. Okay, so moving right along, we now have. Let me back up here. We've now talked about these little gray molecules and the yellow cholesterol. Now we're going to talk about the purple ones. And there's two types of molecules. These are proteins, but the first is called an integral protein, also called a transmembrane protein. And by its name, it implies where it's at, right? Integral means within, and transmembrane means across. So a transmembrane protein would stick out on both sides. Okay? So, how does that work? Well, let me go back. We'll talk a little bit about this when we look at protein formation, but here's my very geometric rough ER, right? In comes that ribosome carrying an mRNA with it, right? And it's starting to produce a protein. Well, that protein is going to be one of a couple things. If that protein is supposed to be part of membrane or secreted, it can do one of three things. First of all, it can be injected into the lumen, and then once it's released, is now in the lumen. Or it can be built and actually fold itself up into the membrane itself, just like that. All right? And getting back to the whole idea of nonpolar and polar, if you have nonpolar R groups on your amino acids, where is it going to be attracted? To the core of the membrane. If you have polar R groups, where is that going to end up? On the ends. So it all has to do with what? It's not, see, this is not magical. It's just, it's a bit more complex than we can really wrap our brains around, but this protein is simply being produced, and if it's nonpolar, it's going to be attracted to the core. So it just keeps added. All of a sudden, this nonpolar section, oh, okay, that's outside. Then you start making another nonpolar, and it folds itself back in, attracted to the core, so now you have a loop. And then you just keep looping and looping until it keeps folding itself backwards and forwards until you have this protein that's exposed on this end and that end with a lot of other structures within the nonpolar core of that membrane. Yes?
What's that A helix?
It's alpha helix. Remember, that's the secondary structure, where primary structure is amino acid, amino acid, amino acid. And then you get a localized spiral, that's an alpha helix. And they tend to be alpha helices with the R groups sticking off the outside. So it looks kind of like a wire brush with the R groups all sticking out like this. And if they're all nonpolar, they're attracted to that middle core of the membrane.
So the alpha helix here is nonpolar and the loops are polar.
In essence, yes.
But it's all one strand.
It's all one strand. Yep, it folds itself up. Imagine if you had like a strand of magnets, and the magnets would be attracted to their opposite, their polar opposite. In this case, it's that idea where if you have a nonpolar R group, it's attracted to the core of the membrane, and if you have a polar R group, it's attracted to the outside, and it just will start to fold itself up. All right, now, once you have this in place, we'll talk about function in just a little bit, okay? But there's one other kind of protein. These are called integral or transmembrane. And if they're sticking out on both sides, there's probably a reason for that. The other kind is called peripheral. A peripheral membrane or a peripheral protein is just that. What does peripheral mean? Yeah, like your peripheral vision's out here. So this is just a protein that's stuck to the side. It's not in the membrane, it's on the membrane. Okay? So we see a couple of them here. We see some down here. These are stuck to, this one's actually a peripheral stuck to an integral. And this is a peripheral that's stuck to membrane. And here's another one stuck to a transmembrane. Here's another one stuck to a transmembrane. But if you notice, a lot of them are attached to elements of the cytoskeleton. Same thing out here. A lot of these are attached to the matrix. So A peripheral protein is just that. It's not going to be exposed across the whole membrane, just on one side. And again, they have various functions. All right, so let's take a look. This is a list of protein functions. And it's very similar to the list that we did with the, when we looked at the macromolecules. And it just so happens that these proteins, as part of membrane proteins, have all this, those functions available to them as well. Some may be for transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and then attachment either internally to the cytoskeleton or externally to the matrix. Now, here's some examples of each of them. First of all, what do we mean by transport? Think about it. If you are exposed on one side of the membrane and the other, doesn't that sound like a window? Sometimes they act that way. In this case, this would be a channel protein, this would be a carrier protein. A channel protein is just that, it's a window. It allows for movement of molecules through the membrane that might not necessarily make it through that membrane anyway. In fact, there's a protein called an aquaporin. I bet you can figure out what that's going to carry, right? Water. Yep, it's a pore for water. In fact, there's so many aquaporins that water passes across the membrane as if it was diffusing. It's like 3 billion a second or something like that. They're very rapid. Sometimes we can actually carry stuff off against the concentration gradient. We can actually concentrate molecules with transport proteins. Sometimes they simply act as an enzyme. They just happen to be linked. When we look at metabolism, we're going to look at something called the electron transport system. In the electron transport system, now it's still membranous. It's on the inner membrane of the mitochondria, and it's literally just an enzyme chain that passes electrons. So I donate the electron to the system. It goes to enzyme 1, we pass it to enzyme 2, we pass it to enzyme 3, we pass it to enzyme 4, and so on. And in the meantime, they're stealing energy away from this thing to make ATP. So that's one way. Another is signal let me just hold on. Yeah, that's another one. It's kind of neat. Signal transduction. It means sending a signal across the membrane. Now, as I said before, steroid hormones, are they impeded by membranes usually? No, because they're nonpolar, which means they will shoot through that nonpolar core just like that. However, let's say a protein, the hormone is a protein, or maybe a complex glycolipid or a complex proteoglycan or something like that, where it's part protein, part sugar, any number of different things. But let's just say it can't cross the membrane. It still has to send the signal, right? So what does it do? It will find this, which is called the receptor, and it's a lot like a keyhole. It's the right shape for that signal, right? But by binding to that receptor, okay, what happens is it's kind of like this. When this one happens, the other side changes. So now, even though the messenger did not go across, the message did. So then what happens is this molecule down here could start a cascade of another series of reactions. So even though the messenger is still outside, whoops, where are we at here? Yeah, here we go. So even though the messenger may still be outside of the cell, the message gets sent. Okay? That's signal transduction. That's kind of how non-steroid hormones work, because they can't pass directly through. Then we've got this type of receptor situation where this protein recognizes this glycoprotein, and if it doesn't, it may attack. This is kind of how your immune system works. There are a number of situations like this, where this could be a red blood cell. This could be a red blood cell, and this is the antigen for your blood type. Another one, the middle one, E, intercellular joining. We know this by a different name. What else do we know this as? This is an example of something we looked at on Monday. Or Wednesday, sorry, we looked at it on Wednesday. You have a transmembrane protein directly attaching to another transmembrane protein.
Desmosome?
Close. Right idea. But I said if we were looking at it from the inside of the cell, we'd see them all lined up like tight junctions. That's what a tight junction consists of. But we're looking at it from the side. I was saying if we looked at it from inside the cell, all we would see would be this end of the protein. But you notice, if this and this are held tightly together, can anything get between them? No. And if you have them nice and lined up like this, you have a seal all the way across. Okay? So, this is a type of tight junction. And lastly, that's when I said, lastly, it could be, this is the most variable one. It shows attachment to the cytoskeleton, it shows attachment to the extracellular matrix. So there's a lot of things going on with proteins. Okay? So, any of those functions can occur in the membrane itself. All right? So far, so good? All right. Now, the one thing I want you to remember is this sounds like I'm being a hypocrite. Or I'm saying one thing and now I'm saying the exact opposite. I said that membrane is membrane is membrane. The core of all membranes are almost identical, especially like within an animal cell. You have the same phospholipid, same cholesterol. But what else did I say? I said the difference lies in which proteins are present. Also, look at your hand. Is the front of your hand and the back of your hand the same? That means you've got a top and a bottom dorsal ventral, right? Same thing applies to membrane. Membrane will have two different faces. It's like a coin. Heads and tails. Okay? Heads and tails. So, a protein can be through the membrane like this, right? The membrane is here. I can be on either side. But again, I've got a left hand here and a right hand here. The two ends might be different. Remember, it's signal transduction, right? If we go back and look at that, this side of the membrane and this side of the membrane are very different, aren't they? Same thing here. This might be a one-way passage. This has the receptor on the outside, but not on the inside. Okay, so again, each side of the membrane might not necessarily be the same as the other. That's why we say it's asymmetrical. Okay?
So a protein that's part of this membrane moves around throughout the membrane, but does it ever leave the membrane?
I would say no. And in fact, they don't necessarily either flip-flop. Phosphates, phospholipids, on occasion, rarely can jump across. Most of the time, they just move side to side. But otherwise, outside stays outside, inside stays inside. But here's the trick. If you know how these vesicles move around, you'll find out that they will turn themselves inside out, completely inside out. And that's the trick to understanding how vesicles work. It's also a trick to know how proteins know where they're supposed to wind up. Okay? So, they know where they're supposed to line up because of the arrangement of components in the ER and then finding the Golgi. Okay? So, here we have three types of proteins. The three types would be a transmembrane protein, that's X, a peripheral protein, Y, and a secretable protein, Z. Now, in the endoplasmic reticulum, this is where they would be, right? As X was produced, it wove itself backwards and forwards into the membrane. As Y was produced, it was stuck to the inside surface. And as Z was produced, it was cut loose and freed into the lumen. All right? So far, so good? So what we're going to do now is we're going to pinch off each of these into their own vesicle. Okay? And here we go. There we go. I was bored one night. Anyway, I'm an artist at heart. Unfortunately, it doesn't make up for lack of talent. But anyway, we now have three vesicles. Right? We've got a vesicle containing a transmembrane protein, a vesicle containing a peripheral protein, and a vesicle containing a secreted protein, or to be secreted protein.
And do peripheral proteins always stay on the inside, or can they be on the inside? Or
When necessary. This one happens to be on the inside, so I just want to make sure I put it so we can track it. But if it was on the outside, it would wind up in a different position. And it's important, to answer your question, there are probably more on the outside, and I'll explain why once we get to that point, okay? Yes?
Does the ER actually break off like that?
That's called creating a vesicle. They actually pinch off, yes.
They do that?
Yep, those are called transport vesicles. Later on, these things would become something like a lysosome.
The book didn't explain that the vesicles actually come.
Yep, they just pinch off. So you keep making more membrane, and as the end gets, they just pinch off. All right, so now, hold on, got ahead of myself. Here we have the Golgi. Now, when these vesicles join with the Golgi, they don't form like this, do they? That wouldn't make any sense. What they do is, let's say I'm the vesicle and here's the Golgi. You fuse like this, and all of a sudden there's a hole, and you now become part of the Golgi that way, right? The inside ends up on the inside of the Golgi. The outside stays on the outside of the Golgi. So as these, here's where my artwork kind of falls apart. But when these vesicles combine, you see where they went, right? They were on the transmembrane, stays transmembrane, peripheral stays on the inside, and secreted proteins are now in the lumen. Now again, as you were saying with the vesicles, they will eventually be modified here, and again pinch off, all right? And just like they will pinch off, very similar to the way they were formed in the first place. All right? Here's the trick. The Golgi and the rest of the cell know where those things are going, and they will be sent to that location. This one might be adding membrane to another vesicle or another organelle, or the cell membrane. Same thing here and same thing here. This one actually could be a lysosome, couldn't it? Because it contains enzymes, free enzymes. But let's just say this one is mucus. We're producing mucus because mucus is secreted. All right, so now watch what happens. Here we have the cell membrane. So this is inside the cell, this is outside of the cell. Here's where it gets tricky. The same process occurs that happened with the Golgi. They fuse like this and become part of the membrane that way. All that becomes added to whatever membrane they're joining. So when that one joins, when that one joins, okay, now you see what happened in the peripheral one? Does that answer your question? Because they were on the inside of the vesicle, they are now on the outside of the cell, and that's where we see most of our receptor, which would be, and that's why if it was like those signal transducers where you have to have the receptor on the outside, now it really matters which side of the membrane they're on. Okay? So in this case, what do we have now? This is now part of the cell membrane, this is stuck on the outside of the cell membrane, and this has now been secreted, like mucus, or a digestive enzyme, or something like that. Okay? Hold on a second. That's why, when you think about it, the membranes have to be kept clear, which is heads and which is tails, because they're asymmetric.
But they wouldn't want to keep adding membrane to membrane that's already there, because then they would grow and lose surface area. So where does that membrane go? We're
going to get to that, but just as it can add, you can take away. And it's the other way around. Let's say, all right, this is inside the cell membrane. Here I am part of the membrane, right? Doing a good imitation, I thought. And what happens? I can do this. I am now a vesicle, and I've just carried in something from the outside. It's called endocytosis. In which case, you pluck away a little bit of the membrane, cell gets a little smaller. So again, you're always adding and taking away that process. Okay?
Okay.
Any questions? That was a good one. All right. So, we okay with this? 10 minutes ahead of my other class. All right. So this is sort of the review. If you look at this, this one's out of your book, but I think the pictures flip-flop left to right, which I found kind of funny. But anyway, here's the ER. Here's the transport vesicles to the Golgi. They bud off. And in this case, they're looking at 3 different situations. One is the secreted protein, one is a transmembrane protein, and one is a peripheral. And in many cases, peripherals are also receptors, which means they have to be sticking out. Okay? So, and they actually show them up close. You see, like these are the free proteins that are eventually going to be secreted. These here are stuck to the inside here, the inside here, the inside here, the inside here, and when they fuse, they now turn them inside out, and now they're on the outside. And this one here is a transmembrane protein, but it's got a particular end on it that needs to be facing outward. So if it needs to be facing outward here, it's going to be facing inward here, modified there, pinched off here. So now it's in the vesicle, transmembrane, but in the vesicle. And as that vesicle fuses, the inside is turned to the outside, and we now have that protein in the right place. Okay? So again, this is constant, it's dynamic, it's always changing. We add to secrete, we then take away by endocytosis and bring in more stuff. We bring in more fuel from the outside, we take away from the membrane, we can add to the membrane, and so on. Okay? Any question on that? Yes? No? All right. Now just remember, this is a generic description because all the vesicles, all the different organelles have membrane. So we might, let's say we want to make a little extra membrane to add to another organelle. We could do the same thing, right? We could make a piece and have that go out into the cytoplasm, fuse with another membrane and become part of it. And whatever you're carrying with it then goes there. So let's say now this happens to be carrying, well, let's say something like this one up here. Let's say instead of this one being mucus, let's say it is digestive enzyme. It goes through, winds up like this, but stays in here. So now the digestive enzymes are inside this vesicle, waiting to fuse with another, and when it fuses with another, dumps its contents into the joint of the two and digests that new vesicle. All right, so everything is very dynamic. It's very recycling, very green, I guess you'd call it. Where parts are recycled, reused, rebuilt. You make more, take some away, you lose some. Okay. Now, 5 minutes, we'll get a little head start on the other class. The next thing we want to talk about is the job of the membrane. All right. Now, if we know the behavior of the substance itself, which we just went over, and we know that the membrane has to be specific, it has to be careful. It's some things it lets in, other things it doesn't, right? So, this is the list of the way that cells can do that. The top half are called passive. These do not require any energy that isn't already in the system. Okay? For instance, molecules bouncing around at a certain temperature. That's the only energy necessary. So the cell does not have to spend any extra energy for diffusion, for osmosis, or for facilitated diffusion. The bottom one's endocytosis, which includes phagocytosis, pinocytosis, and this, receptor-mediated endocytosis, as well as exocytosis does require energy. Because many instances, that is like going upstream. It's like stacking material against its gradient. Where the ones at the top are like going downhill. The ones at the bottom are like going uphill, right? You got to spend energy to go uphill. You can coast downhill. All right, so just as I said before, membranes have selective permeability. Each cell will have the ability to maintain its own concentrations inside because it determines what can come in and what goes out. And again, what's it based on? Basically, it's the chemistry of the individual molecules. Polar molecules, large molecules, anything with a charge on it are not likely to come through. Small, non-polar molecules shoot through pretty quickly. All right? Now, to answer these two questions, that's what you got to remember. The rule of thumb, kind of the easiest, simplest, most by-the-book, middle-of-the-road description is what can enter a cell? Small and hydrophobic. Hydrocarbons, small lipids, things like that. In fact, hydrocarbons can actually start to dissolve the membrane itself. What cannot? The most obvious is the large polar molecule, like big proteins. But then again, large carbohydrates can't as well because they're polar, right? Anything that would be dissolvable in water, anything that has solubility in water, would not easily move in across the membrane. Yes?
So what is done with the larger molecules if we can't do anything with them in the cell, if they can't get in?
A couple of different things. I mentioned one. We've already mentioned one today. Something that would be there instead of going through the membrane.
Attached.
They could just attach or connect to the outside of the protein. And
That's one thing.
Production.
I talk about transport proteins. Sometimes transport proteins are available. Like I said with aquaporins. An aquaporin is a transport protein for water. It's like an open window. Just water can go through as if it's diffusing. It's that quick.
So
By the way, there's other ones. That whole first list, that's it.
Okay.
Every one of those would be a way to move things across membranes.
So a virus then, it's larger than a molecule. So how does it get into the cell, or does it ever get into the cell?
That's a syringe, right? That's what viruses do. They stick to the outside and literally inject their DNA right through the cell membrane. In fact, that's one of the ways, that was graphic representation, but that's how we found out that DNA is actually the genetic material. They were tracking bacteria, they were tracking viruses that infect bacteria, and found out that everything else except for their DNA got left behind. And the only thing that actually went in was the DNA. So what happens is once the DNA gets in, it tricks the cell into thinking it's supposed to be making new virus particles, remakes the virus particles, the cell disrupts, releases new viruses, and they go and infect others. So yeah, they don't actually go through it. In fact, there's actually micrographs in your book showing the virus particles all along the outside of bacteria. Yeah. Any questions? Any? All right. So yeah, this list right here, though, this